I am a graduate student in Debbie Marx's lab, and today I'm going to be sharing some of the recent work we've been doing in developing generative models of sequences for functional library design. And today specifically, I'm going to be talking about this in the lens of how we can optimize for success in antibody discovery. And so first, briefly, an overview of today's talk. Just first, why do we even need computational methods to accelerate antibody discovery? What does the field look like at this point? Second, I'll describe some work that we have done in designing optimized libraries of diverse antibody sequences by learning from natural repertoires. In addition, I'll talk about some more recent work that we have been doing with fabulous collaborators across many institutions on deconvolving fitness into specific biochemical properties with experimental data sets. And then finally, I'll end with a brief teaser on the work that we are actively developing right now on biased library design for optimizing for desired properties by using both natural and experimental sequences. And so let's just start with why we need computational methods. And first, by taking a step back and looking at the field of protein design, which is an especially exciting area of research because of the potential applications to synthetic biology, biotechnology, and therapeutics. And this field can be broadly categorized into three categories of methods of protein design, one of which is molecular modeling, and people such as David Baker have been extremely successful using these methods to design novel proteins. In addition, you have more experimental methods, such as phage and yeast display and directed evolution, which have really opened the way for protein design in the lab. And as you know, this was a Nobel Prize. In addition, more recently in the 2000s, statistical models of sequences have really come to be successful in designing proteins in labs such as Jung-Eun lab and many others, including our own. And this is partly due to the increasing amount of sequences that are available to us, especially as costs of sequencing gets cheaper. Inside protein design, though, antibody design is especially important. These are particularly sought after for biology and medicine. And as you can see in the plot on the left, the cumulative number of FDA approvals over the years has increased exponentially for antibodies. And this just demonstrates how biologics, and especially antibodies, are the fastest area of growth in therapeutics. On the right, I've included a structure of an antibody just to get us familiar with what they look like, and especially the VH domain, which is responsible for much of the antigen binding. And antibodies are especially useful because they have high sensitivity and high specificity. What I mean is that antibodies can bind to the target with really high affinity, so bind strongly, and bind with very specifically, so limiting the off-target effects, off-target binding. And within antibodies, nanobodies actually especially hold a lot of promise. So antibodies generally have a heavy chain and a light chain. And what llamas and sharks and alpacas, camels, etc. have are these antibodies that only have a heavy chain. They don't have a light chain. And yet they still possess the ability to bind to target antigens using the VH domain here. And if you just take this variable domain of the heavy chain, that is what the nanobody is. And so this nanobody is very small. It's a single domain, which makes it extremely useful for engineering. It makes it easier to work with in the lab, etc. So this is a very promising molecule for therapeutics and molecular biology, etc. So how have people traditionally discovered antibodies? Well, usually you have an antigen of interest. You inject the antigen into your mouse model or your llamas or whatever animals that you are using. You extract the blood and then you isolate the antibodies. But this whole process is extremely time-consuming, expensive, and labor-intensive. So the field has really pivoted now to using phage and yeast display methods. And these yeast and phage display methods have had tremendous amounts of success in recent years. And this is actually what is being used in industry and academia nowadays. High throughput display libraries with screening and selection methods have really accelerated discovery. And so what this consists of is you have a nanobody library, or a library of sequences and nanobodies or antibodies. And you can display these molecules on the outside of yeast cells or phage. And now you have a library of cells expressing antibodies. And then you can expose this library to a target antigen that is bound, that has a fluorescent marker or any other marker with which you can now isolate the cells that have nanobodies that bind to these antigens. And so this is a really rapid way of screening millions to tens of millions of sequences of nanobodies or antibodies at a time against a target antigen. And so the state-of-art comparison for nanobody libraries that is described in the McMahon et al. Paper in 2018 from the Cruz lab, they constructed a nanobody library using site-specific amino acid frequencies at a handful of positions along the CDRs, according to the sequences of nanobodies that had crystal structures. So they were able to modulate the frequencies of amino acids at different positions by looking at the structures in the PDB and modeling after that. And this is great because PDB structures, so nanobodies that have a crystal structure are often have high affinity, are stable enough to have crystal structures, and often at least are not extremely sticky to everything. So these are all good examples of nanobodies that are that have favorable qualities as well as good affinity. But the problem is here that even this library, which is state-of-the-art, contains many non or poorly expressing nanobodies. And I'll get to why this is a problem in just a second. But I want to take a moment to describe the pipeline that is now commonly used across industry and academia alike. And this here, you begin with these large libraries on yeast or phage display systems. And you have sizes for these libraries on the order of 10 to the 8th to 10 to the 12th. And then you have, you do multiple rounds of selection, 3 to 10 rounds of selection for binding to a target antigen, after which now you have a pool of moderate to high affinity binders on the order of hundreds to maybe thousands. Then you sample some of those for low throughput experiments, for KD measurements, for biochemical characterizations, etc. And then you can optimize these sequences with something like affinity maturation, for example, to improve the affinity of these sequences as needed, or to improve the biochemical properties of these sequences. But I want to just mention some of the limitations of this pipeline as it stands today. So as I already mentioned, these large libraries often contain large fractions of poorly expressed, unstable, or non-functional sequences. And you can imagine why that might be a problem in just having a lot of unuseful sequences in the starting library, but also because you're passing this down through all of the experimental steps. And so you might end up with sequences that have high affinity, but other poor biochemical characteristics. Secondly, the experimental burden is high. The identification of candidate binders requires many successive rounds of experimental selection and characterization. And not only does it require, many rounds of affinity selection, oftentimes a lot of these selection experiments require negative selections against unfavorable qualities such as non-specific binding or instability, aggregation, etc. And if binders are not good enough for whatever reason, you need to develop more focused libraries and more experiments are required. And then finally, despite all of these experiments, a strong binder with favorable biochemical properties may still not yet be discovered in this way. And so this brings us to how we can use computational methods, specifically statistical models of sequences, to help alleviate some of this experimental burden. And this brings us to my next point, which is to describe our first body of work in designing optimized libraries of diverse nanobody sequences by learning from natural repertoires. So this work focuses on the very first step of the pipeline. How do we optimize these starting libraries? How do we have smarter libraries with fewer non-functional sequences? Well, our lab and others have been highly successful in developing generative models of sequences from evolution and natural repertoires to learn sequence constraints of stability and fitness to predict structure, protein-protein interactions, disordered proteins and states, and the effects of mutations. So then our first thought here was, when thinking about the problem of nanobodies, is can we just take some of these generative models that we have developed and apply them to nanobody sequences? Well, the short answer is it turns out it's not that simple. And the reason is as follows. Existing generative models aren't applicable to sequences with high variability. And what I mean by high variability is this. Nanobody sequences from naive llamas look like this. And if you can see here in this section of the sequence, which is the CDR3 region, and as well as to a lesser degree the CDRs 1 and 2, these regions are highly diverse in both length diversity and residue composition. So while you can construct an alignment of these sequences, the alignment where the CDR3 lies is actually not that meaningful because it's very difficult to align these highly variable sequences. And this is especially important because the CDR3, along with the other CDRs, are the portions of the nanobody that determine the binding to target antigens. So we really need to be able to learn meaningful things about the CDRs in order to make to generate good nanobodies. And so today I'm going to talk very briefly about the model and some of the validation that we have done. If you're interested in more detail, I encourage you to check out Debbie's MIA talk from a while ago. And so you have the alignment-based models, which were the previous generative models that we as a lab had developed. And now the autoregressive model is not dependent on alignment. It actually decomposes the probability of a sequence into the probability of each residue given the residues that occurred before it. And we parameterize this network using a dilated convolutional neural network. And this just allows us to see further back in a sequence, deal with longer sequences while limiting the number of parameters that we use. And so first, though, before we go on to applying this method to generating new nanobody sequences, we had to test first whether the model was actually learning informative sequence constraints. And so we compared our predictions to 40 deep mutational scan data sets and compared the Spearman correlations of our predictions to the experiments with the Spearman correlations for 4 other mutation effect prediction algorithms, such as Deep Sequence, which is a VAE-based model, Mutation, which is a pairwise POTS, as well as an independent model and an HMM. And so that we saw that it matches state-of-the-art missense mutation prediction. We also now saw that we can predict the effects of large insertions and deletions. And very importantly, we were also able to generally predict some thermostability of a diverse set of nanobody sequences.
Hi, June. It's Alex. Do you mind going back 2 slides to the insertions and deletions? Just went too fast for me.
Sure.
Thanks.
Yeah. So for the insertions and deletions, to varying degrees, you can see for IGBT hydratase, we do choose to use an AUC because it's so bimodal. And there are some limits to the predictions, but does that help? I don't know if you had a specific question.
No, that's great.
Okay,
thanks.
Great. Awesome. And so now that we've validated the model works, we can go ahead and generate a smart library of nanobody sequences. And this work is in bioRxiv and also is soon to be published very soon, and has been done very closely with Andrew Cruz, Connor McMahon, and Ashish Manglik. And so we train our model on 1.2 million llama nanobody sequences from 7 different naive llamas. We train the autoregressive model. We generate 10 to the 7th novel CDR3 sequences. Of those, we select 10 to the 5 diverse sequences, and then we test them experimentally by synthesizing the library for yeast display and testing for expression and binding. And what we saw is that the design library resembles the distribution of biochemical properties of the natural repertoire. So for hydrophobicity and isoelectric point, the two distributions are very similar, and the design library relatively resembles the CDR3 length distributions. Additionally, our design library contains more diversity than the natural repertoire, and we can see this by just the distance between sequences that are included in the design library are for more than the distances that in the natural repertoire. In addition, we're not just generating sequences that we saw in the natural repertoire. So we're not just recapitulating our training sequences, which was encouraging to us. So experimentally, we saw that the designed library contains better expressing nanobodies than the previous state-of-art library. And this previous state-of-art library is the McMahon library from 2018 that I had mentioned previously. And so what I mean by this is as follows. One, we saw that we have higher mean expression. So of the cells that are expressing, we see that in the green distributions, we see a higher mean fluorescence. So cells are expressing more nanobodies on average. In addition, we see fewer poorly expressed nanobodies. So in this region here, you can see that there are fewer cells that have nanobodies that are being poorly expressed. And finally, we see a higher proportion of cells expressing nanobodies overall. And as a proof of principle, we showed that we can use this library, although it's 3 orders of magnitude smaller than these large libraries, than the large McMahon library, that it can be used to select for binding to a target antigen of choice. Here, this was HSA, and we identified a moderate binder for which, if we wanted, we could go ahead and do more affinity maturation experiments to make it into a better binder. And it was important to us that this identified nanobody is diverse from the nearest nanobodies in the llama repertoire. And we did indeed see that the sequence is diverse from its nearest neighbors. And while this work was a great first proof of concept that statistical models of sequences can be usefully applied to design better expressed libraries that can still be used for discovery, there are still some limitations that need to be addressed. One is, if you recall, the length diversity of our model generations were actually smaller on average than the natural distribution. In addition, we would like to include more sequence diversity and somehow compensate for the fact that the overall size of our library is small due to synthesis costs. We are synthesizing exact CDR3 sequences, and the cost of this can get very, very large. In addition, we don't explicitly optimize for any specific biochemical properties such as stability, polyreactivity, and affinity. And this brings me to a very important point, which is that our model and our designs are only as good as our training data. And so if naive sequences are the hallmark of very stable, very non-polyreactive sequences, then this is what we would expect to learn. And this is something that we are digging into more as well. So for the first step to improve this V1 design, we went ahead and tried to improve the first few points as follows. So we have worked with a lab down at UC Irvine, Cheng Lu's lab, to integrate our library into an in vivo evolution experimental system. And we have increased CDR3 length diversity. In addition, we've explicitly maximized the diversity of sequences. We've included framework diversity and framework CDR3 dependence, and we optimized the codons for missense mutation likelihood. What I mean is here. So we integrate these design libraries into Orthorep, a system developed in Cheng Lu's lab. And this system allows for directed evolution of genes in vivo. So you can just, there's an error-prone polymerase that targets your gene of interest and can basically search the local fitness landscape around the gene. And if you're interested in this, you can also check out Cheng's MIA talk from, I think, a year or so ago, maybe a little more. In addition, combining the phage display and directed evolution systems into a system called AHEAD, Chang Liu and Andrew Cruz have developed a system such that we can express our nanobody library on the surface of yeast cells combined with this in vivo evolution system. And so what happens here is now, in addition to phage and yeast display and statistical models, we can harness the power of directed evolution for protein design. But in vivo evolution can really only go so far. And so we still need to increase some of the diversity and optimize the functional and accessible diversity for Orthorep. And so first we begin by increasing the CDR3 lengths by correcting tendencies of our model to generate and select short sequences. So as we tend to cluster our sequences, we actually bias our model for generating shorter sequences, and for selecting, if we select centroids of these clusters, we actually choose the shortest sequences, even though they're the most probable, because they have the most neighbors. The shortest sequence tend to have more neighbors. And so we corrected this by sampling from these clusters, and now we have longer CDR3 lengths that match the naive distribution. In addition, we also achieve greater overall sequence diversity by explicitly just enforcing a minimum distance filter. So a sequence can only be included into the library if it is some minimum distance away. And we can see that this does increase the minimum Levenshtein distance between pairs within our V2 library design. In addition, we include framework diversity. So in the first V1 design, we only had one framework region from which we conditioned the generation of multiple CDR3s. Here now we generate CDR3 sequences based on here, for example, 2 different frameworks, but we really have 7 different frameworks, 6 from the germline genes in llamas, and another that we used from the V1 library design. And then we can take these CDR3 sequences and transplant the generated sequences to the other framework genes. And this is an explicit way of asking how much is the CDR3 sequence space dependent on the framework? And this is an important question because we want to know, the model suggests that sequences generated by conditioning on one framework do prefer that framework over other frameworks. However, this could be from learning on natural sequences. This could be just by a downstream effect of VDJ recombination, but we really want to see how much is this really a stability constraint. And so this will allow us to not only increase the size of our library by 7-fold by doing combinatorics, we can also just test this CDR3 space conditioned on different framework genes. Finally, we optimize the codon usage for Orthorep, with specifically Orthorep in mind. And so we have standard codon optimization using the codon usage table, yeast codon usage table, but we also want to bias for codons that are most likely to lead to a missense mutation. So if a nucleotide substitution occurs, as with Orthorep, we want it to bias the codon such that the nucleotide substitution will most likely lead in a non-synonymous mutation. But again, not all mutations are equal. And what they've seen in Orthorep is that transition mutations are 98% of the mutations seen in Orthorep. And so for this round, we optimize the codons for just single nucleotide substitutions, but in the future, we'll account for multiple mutations and learn from experimental data for specific mutation rates as well. And one of the most exciting things about working with Andrew Cruz and Chang Liu and others is that we can now test a variety of these libraries really head-to-head so that we can compare the combinatorial Acmgm library to our V1 design, to our V2 design, to the natural llama repertoire, and incorporate these systems into AHEAD and test the libraries for binding capacity on a diverse set of antigens and for general fitness characteristics. So we can see when our methods have actually improved the hit rate of using our libraries as compared to the natural repertoire or as compared to a combinatorial library or just between our own designs. But this brings me to this next point, which is there are still more limitations that need to be improved. And this involves stability, polyreactivity, affinity, specific biochemical properties that we have still not addressed because we are training on natural repertoires. And so again, because our model is only as good as our training data, it really matters what we're learning from. And there is more and more evidence that natural repertoires are, for example, slightly polyreactive, such that they can bind to a target, after which with hypermutation, it is the nanobodies or antibodies in organisms are selected for specificity. So this brings us to my next point, which is how can we deconvolve fitness into specific biochemical properties using experiments? And so experimental data sets really allow us to explicitly interrogate each and every biochemical property that we are interested in. And this work is done with a fabulous set of collaborators who I will mention in just a bit. But this work is really focused on these last few steps of the pipeline. So can we computationally reduce the number of experiments that we need to identify both a high affinity binder, but also biochemically favorable nanobodies? Can we reduce the number of selection steps, reduce the types of selections, and minimize the need to do further optimizations down the line, and optimize the success rate of finding a good antibody at the end of the day? And so the goal for each feature that we look at is to, 1, predict a phenotype from a sequence, 2, to learn important sequence constraints, 3, we can design rescue mutations. So if you have an antibody or an antibody that is fantastic in every way, you have a great affinity binder, but it's very sticky to many things, or it's unstable, can we rescue that sequence while trying to keep most of its functionality? And then additionally, we can generate optimized sequences. So libraries of sequences, or just additional examples of sequences that have these features optimized. And so this brings me to this fabulous team of collaborators all in this nanobody research. One feature that we can interrogate experimentally is thermostability. And this is work with the Menglik lab at UCSF. And polyreactivity, which is done with the Cruz lab at HMS and the Ring lab at Yale. Evolvability, which is done with the Liu lab at UC Irvine. And affinity, which is done with the Cruz lab and the Liu labs. And then multiplexed affinity, which is done with the Ring lab. And all of these 5 labs, including our own, have come together to form kind of a large nanobody team. And meeting with them and collaborating with them has really opened doors in terms of what we can answer experimentally and computationally to design better nanobodies and better nanobody libraries and speed up the overall process of nanobody discovery. The general experimental workflow for these experiments are as follows. You have a starting library of sequences, and then you do multiple selection steps with max selections or FAX selections with gates, or specific elution of antigen binding phage, for example. And then you end up with a pool of selected sequences. And the kind of data that we get from these experiments is that we have next generation sequencing, or NGS data, sometimes with recounts and sometimes not, at each stage of the selection process. And so just for example, I want to begin by talking about thermostability. And this thermostability experiment is done with Ashish and Jaho. And what they have developed is an assay that really assesses the thermostability of a nanobody using an antibody that only binds to well-folded nanobodies. And so as you play with the temperature, you can see which nanobodies stay well-folded and maintain their structure because of the binding to this antibody. And then with this gated selection, now what you get is a pool of high fitness or high thermostability sequences and a pool of low fitness or low thermostability sequences. And so now we can train a discriminative model on these sequences to be able to predict the class, so high or low thermostability of any given sequence. But first things first, we need to be rigorous with our trained test splits so that we can see to what degree our supervised models are generalizable. And so the problem is that random splits can result in sequences that are very close in sequence space to training sequences. And so the solution is to cluster sequences and then split them into train and test sets by cluster members. So what I mean is this: if these are clusters 1 through 5 and clusters 1 through 5, in the blue distributions are the distances between nanobodies in the two clusters. And you can see that the blue distribution does eliminate sequences that are very near to each other. So orange is the distances between sequences in the same cluster. And so you can see that by clustering, we can get rid of most of the sequences that are highly similar to the sequences in the training set, for example. And we can also additionally place explicit distance constraints and say we will only take things at a certain minimum distance away from the training sequences. And so now that we have a robust split, we can train a supervised interpretable sparse logistic regression model of CDR sequences. And we see that we can make accurate predictions. So on the left is a model trained on a one-hot representation of the sequence. And on the right is a model trained on a k-mer representation of the sequence. So a k-mer representation meaning that we take the 3-mer, 1-mer, 2-mer compositions of the sequence and construct a feature vector and learn a logistic regression model. And we get decent AUCs of around 0.8. Using more complex neural network architectures, we can actually achieve greater accuracy. For example, a CNN or an FNN, a fully connected network, we can achieve higher AUCs of around 0.86, 0.88, almost to 0.9. And RNNs, et cetera, show similar improvement. And so we see improved performance for accuracy, but we also see decreased interpretability in terms of what we can say about which portions of the sequence are explicitly involved in making the thermostability predictions. And this is something that we are also working on to describe. And using what we learned from thermostability experiments, we can go ahead and also translate these predicted scores to actual biophysical TM measurements. In addition, we want to be able to say, for given a low thermostability sequence, we can design sequence optimizations to improve the thermostability of that sequence. We can also, through these models, identify rules of sequence constraints that determine thermostability. And then we can, as a resource, we will provide a model that, given a sequence, we can output both the predicted score, the translated TM measurement, predicted TM measurement, and suggested mutations to rescue the sequence if necessary. Similarly, with polyreactivity, polyreactivity is a problem because you don't want an antibody that binds great to your antigen, but also binds great to everything else. You want it to be highly selective. And so this work is done with Andrew Cruz, Meredith Skiba, and Eddie Harvey. And what they have been doing with us is to do a FACS selection for binding to PSR. PSR is an insect cell membrane prep reagent, which is commonly used as the standard to eliminate highly sticky nanobody sequences or antibody sequences from your selection experiments. And so after a max selection against polyreactivity, they also do a fast-gated selection, and now you have high fitness, which in this case is low polyreactivity or non-stickiness, and low fitness sequences, which is high polyreactivity or very sticky sequences. And as before, we can train a discriminative model. And so what we see here is that we can learn from NGS data of the FACS selections to predict polyreactivity of unseen sequences. And so here, as before, with a model trained on a one-hot representation, we can see the parameters learned by a logistic regression model. For example, you can see that a methionine at position 8 in CDR1 seems to be highly unfavorable, as in it is oftentimes seen in sequences that have high polyreactivity. Other examples are arginines across the board, across the CDR sequences seem to be more common in sequences that are highly polyreactive, as opposed to aspartates, which seem to be the other way around. Similarly, when we train a model on a 3-mer feature vector, we can see the learned parameters here. And while these parameters, there's 8,420 feature vectors. So with sparsity, it's not that full set, but you still see many different k-mers that really contribute to our polyreactivity predictions. I've also just included here a model trained on byte pair encoding, which is kind of a generalized form of a 3-mer model. But more importantly than just being able to classify sequences into a polyreactive or non-polyreactive set, we want to train the sorts and see if we can accurately rank low throughput quantitative measurements. And we see that we are able to with pretty good Spearman correlations with experiments. And these experiments are negative because experimental binding to PSR means that low polyreactivity is down near 0. Whereas our high fitness scores or low polyreactivity scores are above 0. And so you can see that these are the sequences that are non-sticky. And you can see that we have a good, pretty good ability to rank these sequences. And so with this information, we designed mutations to modulate polyreactivity in sequences. And then we're testing these predictions. So for each sequence, we can do an in silico double mutant scan, including missense mutations, deletions, and insertions, and we can select sequences with the largest change in predicted score. So for example, if you take this sequence here, which has low experimental polyreactivity, we can choose mutations that really change the class of this sequence, and we predict to have much higher polyreactivity measurements. And we can do the reverse for a sequence with high polyreactivity measurements, and we can choose something that's near the middle of the polyreactivity experimental measurements and see if we can modulate the sequence in either direction. And we have designed these mutations, and these are currently in the lab, in the Cruise lab for testing. And so we are eagerly waiting those results. But what I really do want to also just mention here in terms of nanobodies is that polyreactivity as a term is a really complex problem. There are many degrees to polyreactivity. Binding to PSR is just one example. For example, you could imagine that a nanobody is polyreactive or just has off-target binding to similar antigen targets, for example, a few different GPCRs. You could also imagine that a sequence could have polyspecific binding to many dissimilar antigens. And then that you can have a sequence that's just extremely nonspecific binding to everything. And so different experimental assays will test different levels. And we're learning to how to describe these different layers and how they compare, and how PSR binding can compare to, these different scales of polyreactivity with some work that we are doing in collaboration with both the Cruise lab and the Ring lab at Yale. So that'll be really exciting to see just how translating an in vitro experiment can really be translated to potentially binding to many, target antigens in the human proteome, for example. And switching gears a little bit, we can also learn about evolvability. So this isn't necessarily an inherent feature of a sequence, such as thermostability or polyreactivity that might be important in organisms, but evolvability in terms of the experimental setup that we have with collaborations with the Orthorep system, the Lou lab, is not all sequences have the same evolvability. And we are working to learn these from neutral drift experiments that Gordon and Alon Wellner are doing, and with and without selection pressure, so we can get these mutation frequencies. And something important for us is that we want to bias sequences for which mutations seen in Orthorep will be most likely to survive. What I mean is this: we want to choose sequences that might lie, for example, on this point on the left plot, just because then as you design new mutations, the local fitness landscape is highly fit, as opposed to if you'd make local mutations and you might drop off in terms of stability and fitness rather quickly, as would be more expected in the right. So using the experimental on neutral drift will actually really help us improve these predictions, because it allows us to focus our search space in terms of the mutations that are likely to occur. And this allows us to limit the number of computations required when we look at the local fitness landscapes. Additionally, affinity is the golden bar for everything with nanobodies. A nanobody is only useful if it has high affinity to a target. And so general steps for affinity are as follows. You have a starting library of sequences, you do n rounds of selection, and you get a selected library. And these are examples of there are examples of this in published data sets, and this is just the general routine panning experiments that are done. Panning plus Orthorep affinity experiments are slightly different because you can do multiple rounds of selection just as before, but then now you can take this library and do the evolution and selection in one step. So this is a mimic for potentially affinity maturation, or you could just plop the starting library here as we do with our own library and do the evolution and selection, which really combines the panning experiments and the affinity maturation experiments all into one experiment. And so the goal here, this is the ultimate goal, right? To design high affinity nanobodies. And this is the ultimate goal, whether you're using experiments or computational methods. And predicting affinity for a specific target is inherently a supervised task. Repertoires from immunized animals might be the exception because you can use these immunized animal sequences to learn about which sequences were upregulated or were enriched, and you can use these to inform an unsupervised model, although there's a kind of label on the sequences because it has been immunized, which is great. And then, but the objectives for designing high affinity antibodies computationally is to reduce the number of selections required, to rank the top candidates from NGS data, to say these are the sequences that you should go ahead and pursue for further characterizations and further experiments down the line. We can also design even higher affinity antibodies than the ones that they select. So you can see this is the pool of nanobodies, but maybe with these mutations, you can have a higher affinity antibody or nanobody. And we can design a diverse set of antibodies as compared to those experimentally discovered. You can take the general rules of sensitivity or affinity to the target and see if you can design sequences that look different, that have different sequence diversity, but will potentially keep the same affinity to the target. And so one example of this, one preliminary work to a GPCR that we have done and that we continue to do is if you take an affinity experiment where you have recounts for sequences before and after selection, you can categorize these, each sequence as either being depleted or enriched before and after selection. And then you can train a sparse logistic regression on one hot representation of sequences. And then you can learn the parameters, just as we did with polyreactivity and thermostability. You can learn which parameters increase affinity and which parameters decrease affinity. And in collaboration with the Mangler lab, we have seen initial evidence that the experiments do show that there is increased affinity when you use these residues, these mutations that we predict to have increased affinity when you compare it to just experimental methods alone. And then on top of that, an especially exciting affinity experiment is the multiplexed affinity experimental setup that Aaron Ring and Fei-Mei Liu have developed in their lab. And this is a pooled enrichment for a set of antigens. So you have a pooled selection and then an arrayed selection to get nanobodies or antibodies that bind to individual antigens of that set of that group of antigens that were used to do the pooled enrichment. And the great thing about this is now you can do multiple selections in parallel. So instead of just having to do one antigen at a time, you can do up to 96 potential antigens at a time, or however many, depending on what you choose to do. And the great thing about this is that we can hope to learn general properties of high affinity nanobodies. And so now we have millions of sequences that have only been seen to bind to one or a few antigens with significant recounts. Another great thing about this multiplexed affinity setup is not only do you have parallel affinity experiments, but you can now compare and contrast across these different experiments when you array have the arrayed selections, because now you can see the specificity of each nanobody for the antigen of interest. So it's a very great way of combining both specificity and affinity in a high throughput manner. And so now you have these sequences that are examples of great nanobodies with high capacity to bind target antigens. And as a thought exercise, one question that we are also asking, and because this will have downstream effects on how we train our models, is are the nanobodies that bind to specific antigens in distinct clusters like the left, or are they more dispersed throughout sequence space? So this is something that we are working to answer and will have an effect on how we train our models. In addition, we're not only tied to experimental data sets, But we can also use public data sets of high quality sequences. And these sequences are examples of good vetted sequences that have high stability and affinity. And we can use this to shape the natural repertoire distribution on which we train to target sequences that are most likely to be developable or highly stable and have good favorable biochemical properties. And this is something we have done. And finally, this just brings me to the very brief teaser on what we are working on now to combine both natural and experimental sequences to have biased library design to optimize for desired biochemical properties. And so the goal here is that we want to bring it all together. We want to design a starting library that would optimize the entire process, not only just to improve our chances, but to optimize to limit the number of selections that are needed because we've already taken care of that in the starting set of sequences. And so we need to learn from both natural repertoires and experimental data sets. The advantage of natural repertoires is the large sequence diversity and the many, many examples of generally fit sequences with a base level of stability and function. And the advantage of experimental data sets is that we can explicitly optimize or select for individual features. And along with that, we have ground truth examples, ground truth measurements of high affinity nanobodies and their biochemical features. And so how we are working to do this is using a semi-supervised method for biased library design. What we need at the end of the day is the probability of a sequence given a phenotype with which we can use to generate new sequences that have the desired phenotype. Well, the great news is now we can learn the probability of a sequence by using unsupervised training on natural repertoires. So this is learning the P of X given phi. And we can learn this from natural sequences using a model like the autoregressive model that we have or any other generative model of our choice. And for the supervised side, we can learn the probability of a phenotype given the sequence, or P of Y given X, by supervised training on experimental data sets. So this is with either logistic regression or any neural network architecture or any supervised model. And so at the end of the day, what we want using this method now is we have these large libraries and we can accelerate the entire pipeline and optimize this antibody discovery process for success using both computational methods and experimental methods alike. So with that, I just want to thank our collaborators, Ashish, Andrew, Aaron, Ring, Cheng Liu, and all of their labs, Tim Springer at the IPI, of course, Debbie, a fantastic mentor, Adam, who is now an alumni of our lab, Ada, Aaron, and Eli for all the work that they've also put into this project as well.
We're very excited to be contributing to this amazing seminar series. So it's very exciting to be talking about our work today. So this work was done in collaboration with, well, Davey Marks in our lab, obviously, and Kelly Brook, who's also a postdoc in our lab, as well as the machine learning group at Oxford University of Yaren Gal. So we also worked with Aidan Gomez, who's a PhD student, and in particular with Pascal Notin, with whom we work very closely in this work. A quick back of the envelope calculation can give us a notion of the huge magnitude of human genetic variants. So if we take a mutation rate of 10 to the minus 8, we can see that each person harbors about 60 de novo variants. And this means that somewhere in our huge population of 8 billion people, we can expect all, except for the very lethal ones, variants to exist at least once. Now, the hope is that we should be able to map this genetic variation to the human disease phenotype such that as sequencing is becoming more widespread and more easily available, human genetic data can be used in the clinic to inform clinical decisions such as diagnosis and preventive care. And while there is the hope of starting to use genetic data in the near-ish future, we are still in the massive infancy of understanding how genetic variation impacts human disease. So if we just massively reduce the problem to just coding regions, and even more to just single amino acid variants. Even on this massively reduced problem, we can see that we still have a very, very small and limited understanding of what is going on. So if we look just as an idea of the numbers, if you look at the number of missense variants in NOMAD, which are about 6.3 million, of those, only 80,000 have clinical interpretation in CleanVar. So CleanVar basically is a repository of classification of clinical interpretation. So basically variants are classified as either benign, likely benign, pathogenic, or likely pathogenic. So you see it's a tiny, tiny fraction, and about 99% or less of the missense variants actually are called variants of unknown function. And this ratio of variants of a known function is only going to increase if the rate at which we sequence people goes faster than the rate at which we determine clinical interpretation.
Yeah, and actually, interestingly, so if you can restrict the problem even further, so even if we just look at genes that have already been associated with disease, so genes where we know variants can give rise to disease, even for that subset, the numbers don't change that massively. It's still only a few % of variants that have been classified to date.
Yeah, right. Not only that, but the labels that we do have are extremely biased, both to some genes because they're genes that are highly studied and more focused on in research, but also bias with respect to population because we know that people that have been sequenced are extremely biased in certain ethnic backgrounds. So currently, this classification of clinical interpretation is performed by combining orthogonal sources of evidence. For example, familial and population genetic studies, and also some high-throughput functional assays are being more and more used, and we'll come back to it later in the talk. And there are different frameworks to try to unify this process of combining orthogonal sources of evidence to try to establish community guidelines to do this in an homogeneous way. One such example of these guidelines is the American College of Molecular Genetics, so the Acmgm Criteria, and that's the one that we'll be discussing here today, but there's more of this nature. So the question is, how can we perform classification at scale? And what I mean at scale is to actually do it such that we can targets these large numbers of variation that I just talked about. Well, we need scalable independent sources of evidence. And this is something that we would like to focus our talk today. The question is, can computational tools help in this task of finding scalable sources of evidence? So this is a bird's eye view of the Acmgm criteria. Obviously, I don't expect you to be able to read this, but I just wanted to give a broad view of how it works. So let's focus on, for example, how to classify something as pathogenic. So the Acmgm criteria aims at providing guidelines to classify a variant either as pathogenic, likely pathogenic, benign, likely benign, or just uncertain significance because we don't know. So to classify something as pathogenic, as you can see, you need to combine a very strong source of evidence with a strong source of evidence, or a very strong source of evidence with two moderate sources of evidence, etc., etc. You can see the gist of it. And they provide what they consider to be strong, moderate, supporting sources of evidence. I highlighted here 3 strong sources of evidence. So you can see PS2 refers to familial genetic information, PS3 to well-established, that is also a vague term in itself, well-established functional studies, to understand the impact of variation in gene function, and PS4 refers to population level genetic studies. So all of these 3 sources of evidence are considered strong in these guidelines. And what is interesting is that all the way at the bottom, as the very lowest level of evidence, we find computational evidence. And not only that, but computational evidence is seen here as multiple lines of computational evidence. in concordance can contribute together to give you a what they call supporting, which is the lowest level of evidence for classifying variants. Now, these guidelines actually do say why the computational methods are so low regarded. And the two arguments provided are low accuracy and poor validation. So we can try to understand better why this is the case by looking at the landscape of mutation effect predictors. Now, this is not trying to be by any means a comprehensive picture of the landscape of these models, because there's been a lot of effort in this space and a lot of different tools developed over the years, but at least it gives you an idea of how it has been changing over the years. So these mutation effect predictors were pioneered by SIFT and Polyphen, which continue to be some of the most used tools by clinicians and researchers, etc. Unfortunately, they are regarded as not being sufficiently accurate to use as a strong source of evidence in some classification scheme like the ACMG one. But over the years, there's been many more models developed, a lot of them supervised, so learning, training on the increasing number of clinical labels, but also meta-predictors, which not only learn on the labels themselves, but also learn on, use as features other models using results that basically guarantee that if you combine different models with different strengths and weaknesses, you're guaranteed to do better overall as a meta-predictor. And if you have a quick look at the results, these are ROC curves for different models, you can see that they are reporting fantastic AUCs and better results. So the question is, why does something like the ACMG criteria still regard computational tools as a low source of evidence? And that takes us to the problem of validation.
Sorry to interrupt. It's Alex. What's ground truth here?
These are the clinical labels in ClinVar, which is per se a complicated topic because the labels are not necessarily very reliable because they're very human, you see what I mean? As well as being extremely sparse. But I think that takes us exactly to the point of validation that Johnny is going to discuss now.
Actually, just one minor clarification. So here, the ground truth is not the same in all of these plots. Primate AI actually does a number of quite clever things. So they use a few different benchmarks, which we won't go into, but generally speaking, or for the purposes of this talk, we're going to focus on using labels from ClinVar as validation. So as we see it, yeah, as Mafalda said, I think probably the key issue here is the validation. And we can kind of break up the issue into sort of three broad categories. Challenges, which we think are actually just a very general set of issues when it comes to trying to make supervised models trained on sequence data, so biological sequence data. But then there's also the class of problems which are specific to the task at hand, which is predicting clinical labels. And then there's a few additional challenges faced by these more recent methods of meta predictors and methods more generally that try to combine multiple sources of evidence. So in the sort of first class of issues, it's really a question of, well, how independent is your test set? And we kind of got a flavor of this in June's talk as well. So, you know, ideally, when we do test train splits for supervised models, we were imagining that the data is independent and identically distributed, but this is not really the case. And when we come to trying to look at sequence data, we can sort of ask for different levels of independence. It's a rather sort of vague process. So, you know, instead of just saying a random test train split, we can say, well, actually, if I've seen that hydrophobic amino acids at position 10 are highly deleterious, then, you know, it's obviously quite easy to predict the effect of other hydrophobic amino acids at the same position. So maybe as a better test of the generalizability of the method, you might want to do your test train split to avoid having in your test set any variance at positions that were seen during training. However, you could take it even further than that and say, well, actually, I'll only look at variants in my test set that are from proteins that haven't been seen in training. Or you could take it even further than that and say, well, actually, many proteins have very closely related domains. And so I'll only test on protein domains that haven't been seen, whereas nothing similar has been seen during training. And obviously, which is appropriate depends on the problem at hand. But when we're trying to do variant classification, we're trying to do a range of these things. And it's really on the onus of the model builders to show that you have sufficient generalizability to do the task at hand. Now, this problem gets sort of augmented or made worse in the context of predicting clinical labels, because the labels themselves are extremely sparse. I mean, this is precisely the problem we're trying to solve, right? This is the intraside that Mafalda just described. So, there are only a very, very small set of labels. And what's more is that they're quite biased. So there's a lot of history behind where these labels have come from. So some proteins have been studied in enormous detail, while some proteins have not been studied at all. They're also biased in terms of what populations the variant, what evidence at a population level we have. So in particular, there's a lot of bias towards sort of European ancestry. And then on top of that, there's the issue of the labels themselves being of really quite variable quality. So actually, the ACMG tried to account for this. They have a sort of a 5-star rating system to say how good the classification process was. But if you start trying to restrict to only the highest quality labels, then what was already a very sparse set becomes an extremely sparse set, and then also an even more biased set. So you have an additional challenge here of trying to play off how many labels you have versus the quality of those labels. And then finally, moving towards sort of issues to do with these more recent methods, is that if if we've already got a sparse set of labels and they're already biased, then validating a meta predictor can quickly become quite challenging if you're not careful, because not only do you have to be careful about the test train splits for all the reasons we just described, but you also have to be aware of the test train splits that were used on each of the models that were used as features. So again, there's yet more pruning to be done. So the remaining set that you can validate on is getting really potentially very small at this stage. And then there's a slightly different issue, which is not meta predictors per se, but just an issue with combining multiple sources of evidence. So one way, of course, to get a better predictor is to draw on very distinct types of training data. In particular, we're going to be talking a lot about evolutionary data, but if you combine that with, say, population data, then you can almost certainly make a better model. But the problem then is that population data is its own Acmg criteria. So if you build a model that trains on that, then it no longer makes sense. I mean, it's no longer sort of compatible with the philosophy of the Acmg criteria or any of these classification processes where you're trying to combine orthogonal sources of evidence, because that's no longer the case. So while you may be able to make a better predictor this way, it comes at a very serious cost of removing other sources of evidence from your sort of classification pipeline, as it were.
Yeah, in a sense, this criteria can be thought of as a meta predictor in themselves, right?
Yeah. So it's not a fundamental issue. I mean, you could think about very nice hierarchical ways to do this, but sort of out of the box, this is not something straightforward to do with something like the guidelines that Mafalda showed before. So with these sort of issues in mind, we wondered about, well, going back to kind of where this field started, which is unsupervised models trained purely on evolutionary data, so looking at the sequences from diverse organisms. And in this way, we've got a model that is unsupervised and therefore bypasses many of the issues with validation that we've described, or at least sort of maximizes the options for validation. It's trained on just a single source of evidence, so it's clearly independent of other criteria in the Acmg, for instance. A key characteristic that we're going to need is that it's scalable, because that's the problem we're trying to solve. And then finally, maybe probably the trickiest part of this talk is discussing how to decide if it's sufficiently predictive to be regarded as something stronger than just supporting evidence. So ideally, strong evidence or at least moderate evidence. Now, fortunately, this brings us pretty squarely to what is now kind of becoming the bread and butter of the Marks lab. So June just gave us a very nice overview. And there's actually now been quite a few methods developed in the lab based around the idea that for a given problem, we collect sequences, in this case, multiple sequence alignments relating to some sequence of interest. And then we train a model to fit the distribution of these sequences. And then we make this sort of assumption that under the model, sequences that are less probable will correspond to sequences that cause some sort of loss of fitness. So in the context of this work, the hypothesis is that improbable sequences under the model will be more likely to be contain disease-causing variants. For this work, we're going to focus on one of the, well, not old methods, but an older method, which is using VAEs that was developed by Adam and John, who used to be in the lab. And this is basically because it's now sort of fairly mature method in the scheme of the tools in our lab, and it's got a lot of very appealing properties for this problem. However, I think in a few weeks you're going to be seeing some very exciting work from Eli, who's been working closely with the Pyro team at the Broad. So yeah, lots of interesting new developments on the way too. So yeah, to give a quick overview of the VAE. One way to think of it is as basically a very efficient way of implementing the following model, which is a latent variable model with a continuous latent space. And the basic motivation for using this kind of modeling is that if you think of a multiple sequence alignment, a typical protein of interest here for us at least is hundreds or even thousands of amino acids long. So if you think of all the, and the possible number of configurations of this, of a discrete sequence of that length goes exponentially in the sequence length. So almost regardless of how, if you've got an amino acid sequence of hundreds and our multiple sequence alignment is tens of thousands or even hundreds of thousands of sequences, that's still an absolutely tiny fraction of the state space of the sequence. So we're training on extremely sparse data when you think of it that way. So the basic philosophy with this kind of modeling is to say, well, actually, if we recast the problem in terms of some continuous latent space, then we gain some notion of distance between sequences, and this can help us to overcome this issue of sparsity. So the sort of generative story that a VAE is basically trying to fit is shown here. So the idea is that you've got some latent variables, Z, drawn from some normal distribution, and this gets passed through some nonlinear functions F, which then get normalized, the normalization is absorbed into F in this way I've written it, to then become the parameters of a categorical distribution, which is how you envisage the sequences we observe being sampled. So to give some intuition for what this means, if we take the special case where A becomes independent of Z, then that means that the model just reduces to a categorical distribution that's statistically independent at each site. And this is quite nice because this is basically what the original, these original models were. So things like SIFT, yeah. So in one way to think of these models is that they're generalizing these previous methods. Now, if we think about a linear F, then what we have is basically categorical PCA up to a softmax term. But then what we have in practice is actually a 3-layer fully connected neural network, which is, of course, a very general function approximator. So what we have now is a framework which allows us to capture a very, very rich set of distributions and generalizes these older methods, basically. So once we have this, as we said before, the idea is that you look at the relative fitness of some reference sequence relative to some reference sequence. And the hypothesis is that improbable sequences under the model should correspond to sequences more likely to be disease-causing. Now, this takes us kind of 90% of the way, but it's not quite enough. what we need for the purpose of variant classification is an interpretable score, and these evolutionary indices are not sort of out of the box, obviously interpretable.
Ideally, we would like to map this evolutionary index to some notion of a pathogenicity score.
Yeah, exactly. So some sort of interpretable score. And when we started this project, we honestly didn't think there was going to be a way to do this in any unsupervised way. We were assuming that we were going to take a semi-supervised approach. However, that changed when we noticed basically this plot that we have here. So just as an experiment, we took the known labels in 1,000 different proteins and plotted what each of these data points is the mean pathogenic and 1 sigma error bar, and the mean benign and 1 sigma error bar for each of these proteins. And what we see is that actually these scores that come from the VAE are somewhat consistent across proteins. It's not perfect, obviously, but I mean, at least to us, it was a surprise. We were not expecting that there to be anything like this kind of consistency. So what I mean by this is that, the value 10 has, very likely means that the variant is pathogenic, and the value 0 means that the variant is very likely to be benign. And this basically motivated us to take a Gaussian mixture model approach. Now, I need to be a bit careful here because this next step certainly doesn't follow from this. It was just that this was the motivation. So what we did next was to take all possible single amino acid variants of a protein and just plot the distribution of those. So it's important to clarify that this has nothing to do with the labeled variants shown at the top here. And it has nothing to do with what variants have actually been seen in a human. It's just the exhaustive scan of single amino acid substitutions. And what we find, actually, quite surprisingly, is that this bimodality seems to persist. So not universally, but taking examples like P10 and SCN5A, there is some bimodality in the distribution of these scores. And so this naturally allows us to fit a Gaussian mixture model. However, there's also examples where this is not the case. So like P53 here seems to have a sort of more unimodal distribution. Nevertheless, you can still fit a two-component mixture model to this. In practice, what we did is we did a mixture of Gaussian mixture models. So we took all the scores from all the VAEs across all the proteins that we studied, which was 1,081 in this initial exploration. And we made a mixture model first over all scores. And then we made a second mixture model that was protein specific, and then combined the two to get what we call a sort of pathogenicity score. And what we get from this is a score which has all the properties we need. So it's a score that runs from 0 to 1, and where 0 has the interpretation of being benign with extremely high confidence, and 1 has the interpretation of being pathogenic with extremely high confidence. And of being a Gaussian mixture model, we also get class assignment. So we get the idea, we get a notion of, we believe it belongs to the Gaussian, to the pathogenic cluster or the benign cluster. And what will be particularly important in what follows is we also get a notion of uncertainty. So we get the idea that this is a high confidence pathogenic or a low confidence pathogenic variant.
Right. So once we combine these step 1 and step 2, we get what we called our model Eve. And we applied it to, like Johnny said, about 1,000 proteins involved in disease. And our results, so our first experiment was to check its performance using AUCs. And I think that this last point here shows well the overall performance. So this is the AUC, an average of the AUC over all the proteins that we studied. And having an AUC of about 0.93, we can see that evolutionary evidence is a strong predictor of pathogenicity. So here in this plot, we show a subset of proteins. These are actually genes that are called actionable genes by the ACMG. They are particularly clinically interesting genes just because their variants are ACMG recommends their variants to be reported just because variants in these genes might be associated with disease for which there are clinical actions that you can take. For example, increase the frequency at which you do breast cancer scans, or change of diet, or put a pacemaker. So very different levels of strength in actions, so to say. And we can see that, that the model performs well across the board. But just like Johnny said, because we have a notion of uncertainty, we can do better than perform classification of all variants. So in the end, what we can do is that if we want to reach a certain level of accuracy, we can do a playoff of how high accuracy we want to get versus how many variants we would like to be able to classify. So if I only classify my most certain variants, I can get very high accuracy. And for what follows in our results, we are going to use an accuracy of 90%, just because that is usually used by the ACMG as some sort of like a goal for predictor models. And you can see that for an accuracy of 90%, that corresponds more or less to retaining 75% of our most certain variants. So basically excluding the 25% most uncertain. And that corresponds to a higher AUC, which are the dark blue dots here. Another thing that we can see in these plots, which is quite reassuring, but in a sense not very surprising, is that the model does not depend very strongly on the number of labels that each gene has. It's not surprising because the model does not train on the labels themselves, but it's still quite reassuring. So basically what you can see here is that these different plot dots correspond to the subset of genes with at least 3 benign and 3 pathogenic labels, or at least 5 benign and 5 pathogenic labels, etc. So you can see that the results are not very dependent on that. And that's just very reassuring because it just implies that there is generalizability across the board of genes. Right. So when comparing the performance against other state-of-the-art predictors, we also see that our model has state-of-the-art performance. So this is a subtle point here. Like Johnny just described, it is very hard to do a proper test range split and so compute reliably validation on most of these models, these supervised models. So instead of trying to do that in a systematic way, what we decided to do instead was to compute upper bounds of the performance of these models. So basically we just computed AUCs for all variants, including the variants on their training. We didn't attempt to separate training and test. So what this plot cannot be used for is to compare across models like this. For example, here polyphen shows as outperforming MCAP, but that's not necessarily the case just because these are just our upper bounds on their performance. But what we can definitely see from this plot is that our model outperforms their upper bounds, which is really, really reassuring. So basically what we conclude from this is that a single source of evidence, evolutionary information, in unsupervised fashion can have state-of-the-art performance, which is really...
This was something that was a bit of a surprise to us, right? Yeah. So with regard to the goals of this work, it wasn't necessary to get state-of-the-art. I mean, obviously it's a very nice thing to see and it's very reassuring, but precisely because it's unsupervised and because it's trained on a single source of evidence, the appropriate usage of this model versus the meta predictor is a bit different, right? So this is a nice feature, but it wasn't necessary. And, it's surprising because, there are theoretical guarantees behind why meta predictors should do well. So it is particularly surprising that a single source of evidence can be so strong.
Yeah, exactly. And here on the right, there's just a different picture of how these performance performs across the board. So we separated the proteins by the ones that have perfect AUC or very high AUC, or high AUC and low AUC. And we can see that our model has a higher percentage of proteins on these perfect and very high AUC range, and less proteins on this lower AUC range. So not only is it state of the art, but it's also very consistent across the whole protein world, which is very reassuring. Do you want to?
Right. So going back to this fourth point of sufficiently predicted to be regarded as strong evidence. I mean, in some ways, this is not something for us to decide. I mean, this is probably something for the community to decide, of clinicians, people using these models to classify. But what we can obviously try to do is put together a stronger argument for why we think it's, the time is right to reconsider the role of these sorts of models. So what we've just seen is comparisons against computational methods, of course, but since they are themselves considered supporting evidence, it's going to be a hard sell to say alone out of from gains of performance there that is sort of time to upgrade these methods. So the other type of comparison we did was against deep mutational scans. And the basic idea here is that they're trying to do something extremely similar to us, right? So as Mafalda mentioned in the criteria, functional assays, well-established ones at least, can be used as a strong source of evidence. So the strategy sort of being put forward by this community is to say, well, actually, we could do variant classification at scale if we can learn to do reliable functional assays at scale. And this is a very exciting field that's seen really rapid progress in the last few years. It's growing enormously. It's seeing enormous financial investment. So a lot of exciting things happening there. And while it's certainly premature, I mean, certainly you can't say that these methods are universally reliable enough to be considered as strong evidence. That's definitely not true. The assays vary enormously, for instance, the relevance of the assay to fitness or to health varies enormously. But nevertheless, there have been some really quite striking successes. So, one very nice example is the saturation mutagenesis experiment by Greg Finlay et al. When he was back in Jason Jura's lab. And they basically show that they get a superb AUC, so their validation is actually just the same as ours. And since then, this data has been used as a source for classification, source of evidence for classification. So the basic reasoning we take is that, well, this field is seeing really rapid investment because there's a great hope of this being used as a widespread source of strong evidence. So if we can demonstrate comparable performance, then it stands to reason that we're bringing an additional equally good quality source of evidence to this problem. You wanted to talk about this one, right?
Yeah. Okay. So when we compare deep mutational scans that have been performed specifically to determine the clinical impact of variants, so specific functional assays determined built for this problem specifically, we see that we also outperform or perform similarly to these experiments. But instead of just showing you yet another plot with AUCs, we decided to just dig deeper into a couple of examples just to understand the nuances and how experiments and models can basically work together on this classification process. So the first example is MSH2. So MSH2 is with MSH6 forming a mismatch repair complex. It's extremely important complex associated with lots of different cancers, in particularly to Lynch syndrome, and is a heavily studied gene. As you can see by the number of labels. So here, the red dots and the blue dots correspond to non-clinical labels in ClinVar, and all of these crosses correspond to variants of a non-function, so variants already seen in the population that either the model on the top or the experiment on the bottom have been able to cover. And here, these lines correspond to our 75% most certain variants being classified, such that the blue crosses are what the model considers a benign or predicts as being a benign variant, and the red crosses are what the model predicts as being a pathogenic variant. It might not be very obvious here on the from this plot, but according to these labels, or using these labels as ground truth, the model slightly outperforms the experiment as in terms of AUC. But when in the MSH2 paper of Jai et al., using their definition of ground truth, which is slightly different subset of clinical labels, they report that the experiment outperforms slightly the model. So in a sense, I think that what is fairer to say is that we are reaching the limitations of using AUCs as a metric, and that both model and experiment are equivalent performance. It's interesting to see a couple of subtle points when we use, or when you compare model against experiment. So for example, both model and experiment call benign labels that variants that are called as pathogenic in ClinVar. So it may you may able to question if the labels are just incorrect, or if the model and the experiment are both missing something. In this particular case, we think that these might be splice disrupting variants, so something that neither model, neither experiment are capturing, and therefore something that is missing on these modeling efforts, and something that we're trying to include now in our model. And conversely, there's also variants that both model and experiment call as pathogenic, but that are labeled as benign in ClinVar. And you can ask if maybe these labels are incorrect. And indeed, some of these labels are already being under reviewed. So some other specialist cohorts already thinks that these labels are incorrect. So you can see that model and experiment can be used in conjunction to try to ask things about the reliability of labels as well. So that's really interesting.
And this is something that would have been hard, if not impossible, to do with a supervised approach. So it's, and indeed, we've done this for a few examples now, and it does seem to be that we can detect potentially incorrect labels. So yeah, this is something we're also looking into in more detail now.
Yeah. Once we plot the continuous Eve scores on the 3D structure of this complex, what we see is that, not totally surprising, the hotspots of pathogenicity are on active sites like ligand binding sites. Here, the strongest pathogenicity hotspots are on the ADP binding site and on the DNA binding site. What was really surprising for us is that some of the highest pathogenicity variants in our model were actually variants that have been seen in the population and had no clinical interpretation in ClinVar. And that basically just tells you that, well, it's just an example of how sparse our knowledge currently is, that even on a gene like MSH2 that is so heavily studied, there's still things that the model can say are extremely pathogenic that are not currently known as such. So another example that is very interesting are ion channels. So ion channels are also very studied because they're involved in loads of different disruptions, from cardiac disease to neurological disease to muscular disease. It's a very interesting subset of genes, and very hard to study in a high throughput functional manner. Here we show a study that basically was pioneered by Glaser et al. Tried to make a high throughput experiment of patch clamp experiments of SCN5A, which is involved in lots of different cardiac disease. And as you can see, the number of variants that were studied in this experiment is much sparser than something like this functional assay. But still, this is just like really inspiring, pioneering work on trying to do this type of experiments in a high throughput manner. So the first thing you can immediately see is that the model can help the experiment by basically having a much higher coverage and a higher scope, and basically can also help, in principle, the model, the experiments direct their experimental efforts to something that might be particularly relevant for clinical setting. Another interesting point is that some variants, for example, this one, were in the experiment picked up as benign, but even on the experiment, the authors suggested that maybe this was a gain of function case. And in fact, they suspected that this variant should be pathogenic due to gain of function. And indeed, we see that in the model, because the model does not really distinguish if it's loss of function or gain of function. In the model, this variant comes as pathogenic. So it's another example where model and experiment can be combined to inform clinical efforts. And if we plot the if scores into the 3D structure of some ion channels that do not have any functional assay currently to date performed on them, we see again that there is very reliable hotspots of pathogenicity. So this is SCN4A and SCN1B that are bound to each other to form a sodium channel complex. And you can see that where SCN1B binds to SCN4A, there's a hotspot of pathogenicity, as well as the pore of the SCN4A. So,
Right, so this basically brings us more or less to the end of the talk, actually. So what this last bit is kind of an exploration of impact. So what we wanted to ask here,
the logical conclusion.
Yeah. So what we wanted to ask here is, well, if you do kind of believe the case we've made today, that maybe the time is right to upgrade computational models trained on evolutionary data, what kind of impact could that have? And so what we did here is we took the roughly 1,000 proteins in this initial study of ours, and we just performed variant classification according to the Acmg criteria, but now using our model as a new form of strong evidence. And what this Sankey diagram at the bottom shows is kind of how this process looks. So on the left, we have the lay of the land before the analysis. So this is the number of pathogenic labels, so roughly 5,000, likely pathogenic, benign, likely benign, and then the overwhelming majority are variants of uncertain significance. And what we see is that the model actually manages to classify a pretty significant fraction of these. And then in the final step, what we see on the right-hand side is what happens when we combine the evidence here. So this sort of treating this as a strong source of evidence with evidence such as frequency data from NOMAD, things like this, other criteria from the Acmg. And what we see is that actually a pretty significant fraction of the pathogenic variable labels end up being classified as pathogenic, but a far smaller fraction of benign end up being classified as benign. And this is nothing to do with the model. This is not an asymmetry of the model, but an asymmetry of the classification process. That it's basically a statement that it's it's actually much easier to find evidence that a variant is pathogenic than it is to find evidence that a variant is benign. So that's basically all that's being said here. And what we find is that we can actually classify about 4 times more variants. So that comes to something like 72,000 variants of uncertain significance that can be classified if we allow for the model to be treated as a strong source of evidence. And of course, as we scale this to more proteins, the impact will be far, far larger. So yeah, that basically brings us to the end of the talk. So to summarize, the big problem that we're trying to solve here is one small step in the goals of clinically actionable, using genomics in the clinic. In particular, the problem of variants of uncertain significance. So this huge fraction of variants for which there's no interpretation that seems to be growing ever more as sequencing becomes more widespread. Basically, what we've tried to do here is question whether or not it's time to reassess the strength of evidence that's being put forward by evolutionary data, and in particular, what can be done when you use sort of state-of-the-art unsupervised models on this data. I mean, when you think about it, it's, you know, I think coming to biology from physics, this is something that I still find really stunning, actually, that, you know, we can build a computational model that's trained on sequences that can predict the chances of disease in a person because someone went out and sequenced a whale. You know, I still find this amazing, actually. And basically what we're getting from these ever more sort of rich computational models is the ability to learn from ever more diverse organisms. I think that's kind of one way to see the benefits of using the VAE over simpler models, for instance. So basically, when we do this, what we see is that actually, you know, we're finding that these models perform extremely well, and actually, surprisingly, seem to outperform even meta predictors at present, which was a very nice surprise for us. So yeah, we think this is basically a big step towards being able to do variant classification at scale, and we'd love to discuss this with you and hear what you think. Looking to the future, you know, it's kind of clear that this approach is limited. Like, this whole setup is rather limited, and there's a lot to be done. You know, a lot of diseases are not well described as in terms of single variants as being simply benign or pathogenic. You know, perhaps it's better to consider the effect of some variants more on a scale. You know, and you know, I think an example of this would be Brugada syndrome, for instance. And what's nice is that our model naturally provides a continuous score. So one thing we would like to do in the future is think how to make better use of this sort of finer grained prediction process that we have. You know, it's sort of a shame that we take a continuous score and then coarse grain it.
In a sense, it's kind of a statement of the status of the field as it stands right now, that this is how clinical interpretation exists right now as available to a clinician, for example, that is interested in the effect of a given variant. This is binary statements of benign, likely benign, or pathogenic and likely pathogenic is what's the current gold standards, which is very limited. And it would be great to move away from that in the future, and something that we're thinking about very actively.
Yeah, and another thing that we didn't make any use of, which comes kind of for free with this kind of modeling, is that we can take into account the effect of multiple variants at once. So this whole analysis is focused on single amino acid substitutions, but of course we look at the scores of multiple substitutions at once, and this is something we're really excited to look into.
So basically we ignore completely problems such as penetrance and tetrapates.
Yeah, I mean, so yeah, looking sort of further ahead, I mean, you know, surely the future of genomics in medicine is not to try and classify each variant one at a time. You know, at some point we need to start looking at whole genomes and thinking about how you know, of course, how variants act in concert. And I think something we're starting to think about now is what are some sort of tractable steps towards that. You know, one day presumably we'll have whole genome models, and but that seems a little bit distant right now. But we can start to think about what are the intermediate steps. And yeah, that brings us to the answer. Thank you very much.
Thank you.